Dear students, today we will discuss on diseases of crucifers and its management. Cruciferous vegetable group constitutes low volume and high value seed crops. The amount of vegetables produced in the country at present serves per capita per day intake of vegetables only around 145 grams against the requirement of 285 grams. Unfortunately, diseases are the important limiting factors which restrict the fast expansion of cultivation and productivity of vegetable crops. The important diseases of crucifers, symptoms and management practices are discussed here. First important disease is the damping of and wear stem. It is caused by Pythium species, Fusarium species, Rhizoctonia solani. Now let's see the symptoms of the disease. Pre-emergence damping off is generally caused by the invasion of the host by the fungus prior to plant emergence from soil. This is due to conditions that inhibit or slow seed germination while allowing pathogens to grow. Post-emergence damping off occurs on young seedlings at or near the soil line. The host tissue appears water-soaked and constricted, eventually leading to seedlings collapse. Infection on hypocotyl or stem of older plants by Rhizoctonia solani may produce canker. Infected stems may be somewhat smaller in diameter than normal, but tough and wiry, hence the name wire stem. Next, let's see about the epidemiology of the disease. The disease is promoted by high humidity and high temperature. Temperature between 24 to 30 degrees centigrade is congenial for the development of the disease. Oospores survive in the soil, act as the primary source of inoculum. Now coming to the management of the disease. Cultural practices. Fresh seed should be purchased each year. Seed should be planted in the recommended depth. Seed should be always planted in raised seed bed. Use sterilized soil when planting seeds in pots. Disinfect tools and potting containers used in seedling or transplanting operation. Seed treatments with metalaxyl or cymoxanil or thiram or captan at the rate 2.5 gram per kg seed, or mixture of carboxyn and thiram at the rate 3 gram per kg seed. Seed treatments also can be done with biocontrol agents such as trichoderma at the rate 10 gram per kg seed or Pseudomonas fluorescens at the rate 5 gram per kg seed. Next cultural practice is the soil drenching. Soil drenching can be done with chemicals such as metalaxyl plus mancozeb or copper oxychloride at the rate 2 gram per liter of water. Drench the soil 5 to 6 days before sowing and repeat 3 to 4 times at 10 days interval. Now coming to the next important disease, it is the black leg. It is caused by Leptosphaeria maculans. Anamorphic stage is the Fuma lingam. Now coming to the symptoms of the disease. Symptoms manifest as oval, sunken, light brown cankers with purple to black margins near the base of stems. Lesion may also develop on cotyledons and hypocotyls of young seedlings and appear on leaves as pale irregular spots. Leaf spots gradually enlarge, becoming circular with grey center. Now coming to the epidemiology of the disease. The fungus can survive in crop debris and cruciferous weeds. Infected seed, however, may also be a source of primary inoculum. The disease may also spread by splashing rain, workers and equipments. Now coming to the management of the disease. Destruction of the crop residues by rapid plowing in the field. Fumigation, solarization or flooding of infested fields helps in reducing field inoculum levels. Crop rotation with non-host plants for at least 3 years. Hot water treatment of seed at 50 degrees centigrade for 30 minutes can be effective for lightly infected seeds. Benzimidazole fungicides are the most effective as slurry treatment. Next important disease is the club rot. It is caused by Plasmodiphora brassicae. Now let's see the symptoms of the club rot. The most characteristic symptoms of the disease consist of small or large spindle-like spherical club-shaped swelling on the roots and rootlets. Affected plants show decline in vigor first and partially recover by forming adventitious roots. The infected roots may rot and perish as a result of secondary infection. Now let's see about the epidemiology of the disease. Infected roots serves as the major source of inoculum and release zoospores, which infect root tissues. Acidic soils and temperature ranging from 12 to 27 degrees centigrade allow for rapid disease development. Now let's see the management of the disease. Eradicate cruciferous weeds and volunteers. Implement 5 to 7 years crop rotation with non-host. Lime soil to a pH of 7.3 or more. Seedbed areas can be kept free from the pathogen by treating the soil with chloropicrin or formalin approximately 2 weeks before planting. Next disease is the Alternaria leaf spot or Alternaria blight. It is caused by Alternaria brassicae, Alternaria brassicicola and Alternaria raffani. Now let's see the symptoms of the disease. These Alternaria species cause leaf spots that appear on older tissues and often begin as small circular lesions. These lesions expand and develop concentric rings with chlorotic halos. They may also infect seedlings with symptoms appearing as black streaks on cotyledons and hypocotyls, which may result in damping off. Infected pods produce small, discolored and shriveled seeds. Now let's see the epidemiology of the disease. The pathogen survives through spores or mycelium in disease plant debris and weed host like Anagallis arvensis, Convolvulus arvensis, Chenopodium, radish and other cultivated crucifers. Moist, that is above 70% relative humidity, and warm weather, that is 12 to 25 degrees centigrade with intermittent rains play a vital role for disease development. Now coming to the management of the disease. Disease can be managed by crop rotation, burn debris, deep plowing, application of balanced dose of fertilizers, use healthy seeds or pathogen-free seed, removal of weeds, spray with fungicides like mancozeb or iprodion at the rate 0.2% at 15 days interval soon after disease appearance. Next important disease is the white rust. It is caused by Albugo candida. Now let's see the symptoms of the disease. White rust affects every known cruciferous crop. Symptoms manifest as chlorotic or necrotic spots on upper leaf surface. Later pustules form on abaxal leaf surface, small stems and floral parts. Pustules rupture the host epidermis and expose a white chalky dust of sporangia in small zonate areas. Occasionally affected portions of leaves are swollen and distorted. The flower stalks, distorted stems and flowers result in a stake head appearance. Now coming to the epidemiology of the disease. Overwintering of white rust by means of oospores in plant debris and weed host serves as primary source of inoculum. Moist, that is above 75% relative humidity, and cool weather, that is 5 to 12 degrees centigrade, with short days, that is cloudy days, 2 to 6 hours sunshine favors the disease development. Now coming to the management of the disease. Disease can be managed by crop rotation, burn debris, deep plowing, application of balanced dose fertilizers, use healthy seed or pathogen-free seed, seed treatment with metalaxyl at the rate 6 gram per kg, spray with fungicide like Ridomil MZ 72 wettable powder at the rate 0.2% at 15 days interval soon after appearance of the disease. Next important disease is the bacterial soft rot. It is caused by Pactobacterium species Pseudomonas marginalis, Pathova marginalis. Now coming to the symptoms of the bacterial soft rot. Symptoms first appear on leaves as small water-soaked lesions that quickly enlarge. Affected tissues turns brown and becomes soft and mushy with an accompanying foul odor. Eventually leaves, stems and roots may decay entirely. This disease may be found in the field on cabbage, Chinese cabbage, rutabaga and turnip, but post-harvest soft rot during shipping or storage account for the majority of the losses from this pathogen. Now let's see the epidemiology of the disease. Soft rot bacteria survive in soil and decaying plant material and infect plants through wounds, stomata or hydatodes. The pathogen is generally spread by irrigation water, rain, several species of maggot flies and other insects. Disease development is usually favored by warm temperature of 25 to 30 degree centigrade. Now coming to the management of the disease. To minimize soft rot losses, control insects, try to avoid mechanical injury during harvest, packing and shipping, and do not pack the produce when wet. Additionally, store and ship produce at temperature near 4 degree centigrade. Now coming to the next important disease, that is black rot. It is caused by Xanthomonas campestris, Pathova campestris. Now let's see the symptoms of the black rot. Symptoms manifest as localized wilting at leaf margins. Wilted tissues becomes chlorotic and progresses to form the characteristic V-shaped lesions associated with this disease. Within chlorotic tissue, leaf veins turns black, giving the disease its name black rot. At advanced stages, affected tissue becomes brown and necrotic. Now let's see about the epidemiology of the disease. The black rot organism can survive in crop residue for up to 2 years. The bacterium can also infect cruciferous weeds such as pepper grass, wild radish, black mustard, wild turnip and others. Though hydathodes infection is most common, stomatal entry may occur when plants are subjected to heavy rains or irrigation. With warm temperatures of 27 to 30 degrees centigrade, symptoms may appear in 10 to 12 days. Spread of the disease in the field generally occurs by wind-blown rain, irrigation water, cultivation, insects or animals. The bacterium can be seed-borne, which may result in seedling infection. Now coming to the management of the disease. Use high quality seed free of Xanthomonas campestris pathova campestris. Implement a 3-year rotation of non-cruciferous crops. Seed beds should be geographically isolated from commercial crucifer crops. Do not clip transplants. Plant crops in well-drained soils and use irrigation practices that minimize leaf wetness. Keep fields free of cruciferous weeds. Disinfect seed beds and equipments with steam or germicidal spray before use. Control insects to minimize spread of the pathogen. This is about the management of the disease. Next important disease is the yellows. It is caused by Fusarium oxysporum, Forma specialis coglutinans and Fusarium oxysporum, Forma specialis refani. Now let's see the symptoms of the disease. Affected foliage turns dull and chlorotic. Some leaves appear distorted due to uneven growth. Leaves may die prematurely and senescence, starting at the base of the plant. This pathogen invades the vascular system of host plants, turning the vascular tissue brown or yellow. Plants which do not die are often stunted and have one-sided yellowing of the leaves or stem. Now coming to the epidemiology of the disease. This fungus survives in the soil and produces spores which can persist in the soil for many years. The fungus enters the plant through the roots and moves into the vascular system. The disease is generally favored by warm temperature. Below 20 degrees centigrade, disease development is greatly reduced. Now let's see about the disease management. Resistant varieties help provide the most effective control of this disease. Next important disease is the cauliflower mosaic. It is caused by cauliflower mosaic virus. Let's see the symptoms of the disease. Only members of the crucifer family are susceptible to CAMV. Systemic symptoms consist of a clearing or chlorosis along the leaf veins. Later symptoms appear as dark green areas along veins and necrotic spotting of the leaf. Chinese cabbage is particularly susceptible to CAMV. In addition to vein clearing, a striking mosaic may develop with light and dark green areas on leaves. Internal necrotic spotting in stored cabbage has been attributed to CAMV infection. Next is the epidemiology of the disease. The primary inoculum source of CAMV is infected brassica crops or cruciferous weeds. The virus is transmitted to the crop by many species of aphids such as cabbage aphid, the false cabbage aphids and green peach aphid. Aphids can acquire and transmit CAMV within 1 minute of feeding on an infected plant. Temperatures between 16 to 20 degrees centigrade favor symptom expression in plants. CAMV is often found as a mixed infection with turnip mosaic virus, resulting in more severe symptoms than when either virus is present alone. Now let's see the management of the disease. Eradicate cruciferous weeds and volunteers, and incorporate crop debris immediately after harvest. Isolate transplant beds from commercial crucifer crops. Use of insecticides for controlling of insects. Now coming to the conclusions of the topic. Cruciferous plants can be protected against diseases by physical, chemical and biological means, or by its own internal barrier known as disease resistance. Important diseases of crucifers are damping off, black leg, club rot, Alternaria leaf blight, white rust, yellows and cauliflower mosaic. The present topic discussed on symptomatology, epidemiology and probable management of some important disease strategies with reference to pertaining original research work. The use of resistant variety is one of the best way to control certain diseases.